similar to it. And I also like to thank the organizers for giving us the opportunity to deliver a session. So moving ahead, as for my today's session, I'll be talking about the role of genetic engineering in crop improvement. So genetic engineering, first of all, and I was seeing many of you were showing queries that you want to listen in Hindi also, or you want us to tell some points in Hindi. I'll try to do my best so that it explains actually the information, what I'm trying to convey. So, talking about genetic engineering, it has a huge revolutionary thing, which especially when we talk about science and when coming down to the field of agriculture, we have also seen a lot of novel techniques which has come up to enhance crop productivity and quality. Basically, evolution, if I say so, because of evolution, we know a lot of changes are happening nearby in the agriculture sector, in the disease sector, in medical sector, and everywhere. So to tackle those changes, we also need some improvisation, some new techniques which will help. So when we say what are the things happening wrong right now, if you talk about crop, agriculture is something which is India's one of the prime sectors, no, for earning. So अगर मैं हिंदी में बात करूं तो भारत की सबसे बड़ी इकोनॉमिकल स्ट्रेंथ है एग्रीकल्चर, जिसमें अब जब एग्रीकल्चर की बात करते हैं तो दो चीजें बहुत इंपॉर्टेंट है, हाउ मच क्रॉप प्रोडक्शन इज हैपनिंग एंड व्हाट इज द क्वालिटी. So some place both the things are getting disturbed. It is because of like बहुत ज़्यादा फर्टिलाइजर, केमिकल्स, पेस्टिसाइड्स एंड ऑल की यूज के वजह से, और environmental calamities. Some are because of man-made actions directly influencing the quality and productivity of the crop, and some are indirectly doing it. Like if we say nothing is directly linked to, if saying that rain. So rain is very important for crops, but many a places we are seeing when we are supposed to expect rain and it's important for the crop, it will not happen that time. And वो तब होता है जब उनको जरूरत नहीं होती है. So again it impacts the crop productivity and quality. So it is important that we deal with such situations, and we are taking the help of technology. Talking here about genetic engineering, it's a technology which is going to help us deal with those type of problems existing, direct or indirect man-made effects. So one of the key aspects of genetic engineering, if we look into the agriculture, is somewhere we are trying to change the genetic makeup. So we cannot change the climate. Can we change? No. Can we change our environment? No. So what we can change? We can change the plant. We can change the crop. So on what basis I'm trying to change it? I'm trying to change it at the level of genes. I hope you all know what genes are. So basically, this is the information which we have in our body, which denotes each one of us. We all are individuals, right? So even crops also have genes. So some genes can be manipulated, या हम उनको थोड़ा सा मॉडिफाई कर सकते हैं ताकि वो हमें कुछ बेनिफिट्स दें. What kind of benefits we are expecting? We are expecting in terms that we need not use the pests. Now how they will do so? They will secrete something. वो कुछ ऐसा प्रोड्यूस करेंगे जिससे pests will run away or pests will not come nearby. So those things will help us deal with the issues. It utilizes the biotechnology tools to introduce some specific properties or traits which we want in our crops. Like, as I said, environment is a huge, like a very important aspect when we talk about agriculture. Like when the uh, rain is going to happen and how much the temperature is going to raise or go down is very important for a crop to survive in that environment. And if a crop is unable to survive, we need to make it survive by doing some manipulations at the level of genes. So, as the term suggests, it's something which involves you changing the gene composition of an organism, and the complete sets of gene is encoded in its DNA that determines its traits. So, what are traits? Basically, if you look into a plant has leaf, fruits, and other things. So, what will be the color of leaf? What will be the color of fruit? Whether the fruit will have a sour or a sweet taste. So, all these information is there in the genes, and that genes are very important for the plants. So, if we can modify that to get a better quality, to get a quality which is not there before, so all these are going to happen at the level of genes. So here scientists basically are working to introduce some desirable characteristics which are not there. So something which is nature has not given them, which we are trying to incorporate. It could be improved resistance. Resistance to what? To pest, कीड़े मकोड़ों से, या resistance to even your climate, you can say. Yield should be better. So instead of having a low, like less crop yield, we are expecting a higher crop yield and a enhanced nutritional value. So many a times we also do biofortification. We incorporate some genes which helps in, targeting malnutrition, something like that. I'll talk about in the future slides about it. So there are some organisms which are developed using these methods and which we term as GMOs. So what are GMOs? It's basically genetically modified organism. I want to make it clear that it is not naturally produced. We are making it. Is it making sense? Audience, are you able to understand? Okay. So there are a few milestones which we have seen when we talk about genetic engineering. The first one was with TMV, tobacco mosaic viruses. So it was giving us genetically engineered plants, which were initially commercialized in China. So what they were resistant to the virus. They were made so that the virus can no longer infect those plants. Now, how we can do it? So, अगर आप वायरस के बारे में जानना चाहोगे तो वायरसेस कुछ स्पेसिफिक सेल्स को बाइंड करता है. उन सेल्स पे कुछ प्रोटीन्स होते हैं जिससे वायरसेस जाके अटैच करते हैं. अगर मैं उन प्रोटीन्स को हटा दूं तो viruses can not no longer go and bind. But when I'm trying to do so, I should also make sure that while removing those proteins, it should not affect the normal functionality of the crop. Okay. So when you're trying to do a genetic engineering, please make sure that it does not affect the nutritional aspect of the crop. That should be kept intact. Okay. So lots of things needs to be taken care. Another thing which came was 1994, where we have some special categories of tomatoes. So tomatoes have a problem that, not a problem, like naturally, uh, normal sense, they ripen very fast. So when we have to export or import tomatoes, it's difficult because by the time it reaches the destination, it is spoiled. So there they had to increase the shelf life. like how I can make sure that the ripening process is slow, जैसे कि वो लेट तक फ्रेश रहे. Okay, so these are very important. I'm trying to say as much as possible in Hindi because I may not know exact terminologies, but I'll try. Since because these are very scientific terms, no, so I may miss out, and I'll try to make it simple. This is not very difficult, which I have made for you people. So that came as where genetic engineering was used to increase the shelf life of the crops, specifically the tomatoes. Next came in 1996 was cotton. So cotton is something which we are in India has also adopted as a GM crop because it's like protection of crop from pests. So there was some warm pests which were spoiling the cottons. So it secreted the modified crop uh, एक ऐसी चीज पदार्थ को प्रोड्यूस करती है जिससे वो पेस्ट आर किल्ड, like they are destroyed, they are removed, they cannot come and spoil the or infect the cotton. So what happens, you get a good quality of cotton. Other than that, we have herbicide tolerant soybeans which were produced in the year 1996. So they were basically used designed to tolerate herbicides which kill the surrounding unwanted plants. So it gives more space. एक प्लांट को आप किसी भी प्लांट को बहुत क्लोजली नहीं ग्रो कर सकते हो. दे नीड सर्टेन स्पेस, बट दिस herbicides and all the weeds, if they start growing nearby, what they do, they damage the normal growth of plant. The plant cannot grow properly. Other than that, we have lots of diseases which we are seeing, like rainbow papaya was being produced. So it gives protection of to papaya from ring spot viruses. Again, the same thing which what we saw in TMV. So like this, here also, we will not allow those proteins to be produced. So proteins कुछ ऐसे molecules होते हैं जो आपके सेल के ऊपर होते हैं. So कोई भी प्लांट या थिंग का एक बेसिक है सेल. सो एक वायरस सेल को इन्फेक्ट करता है और उसके लिए उसको कुछ स्पेसिफिक चीजों से बाइंड करना चाहिए जिसमें वो प्रोटीन्स बहुत इंपॉर्टेंट होते हैं. सो इफ यू रिमूव दोज प्रोटीन्स, व्हाट हैपेंस, दीज वायरसेस कैन नॉट इन्फेक्ट. एज अ रिजल्ट, योर क्रॉप्स आर नॉट डिस्ट्रॉयड. Other than that, we are going to look for the nutrient aspect. As I mentioned, key nutrient is also something which we need to keep in mind because end of the day we are delivering something on a table which needs to be edible and which needs to be nutrient high in nutrient content. So some crops were produced which were high in some specific nutrients. So golden rice was there to boost the vitamin A in the children, and thus it also helped in tackling the vitamin A deficiency. Other than that, recently 2017, we had something called Arctic apple, which controls browning in the flesh of apples after being sliced. So अगर आप सेब को बहुत देर तक छोड़ दो तो आपको दिखेगा कि वो थोड़ा रेडिश ब्राउनिश हो रहा है उसके टॉप की लेयर. So in these GM crops which are genetically engineered, those initial browning is not going to take place because it is being controlled by the help of genetic engineering. So these are the milestones which has been taken care while using genetic engineering in agriculture sector. I'm only talking about agriculture sector. I'm not going into any other sector because I can assure you that this field is the need of the hour because everything is evolving and so do we need to evolve. And we can only evolve when we go with the technologies. Like right now, we have a AI generation. Okay. So if we don't use AI, if we don't go with it, then we will be back end. So now, if I compare the genetic engineering process with the conventional breeding, someone few minutes back talked a lot about breeding sector. So I'll just touch upon it and just show you how they differ from each other, because इनमें कुछ differences हैं. जब आप conventional breeding की बात करते हैं तो उसमें आपको सिर्फ एक trait नहीं मिलता है. Suppose there are two plants. One is having a trait which I want in another plant, but apart from that, it is also having 10 other traits which I don't need it. But still, it is going to be crossed. So apart from one requirement, like one desirable trait, I'm also getting 10 other traits, which I don't want it. So I cannot help. It is not selection. It is not that I can specify, take only one trait. It is difficult in conventional breeding. It is not possible. Whereas in genetic engineering, we can take only a specific trait, try to modify it, and we can see the results. When we say conventional breeding, the process and the precision is very low because there is a mixing of entire genome. As I said, two individual plants are being mixed. They are being breeded together. But when we talk about genetic engineering, it is very high in precision because specific genes are targeted or edited. It is not something undesirable is also going to come with it. So that's what happens. No, if you see your parents, like you have a maternal side and a paternal side, so you get traits from both. You cannot select it. Not possible. That's what happens in breeding. But when you talk about genetic engineering, you can specifically see, oh no, I only want brown eyes. I only want yellow color fruits. That is what precision talks about. So that part can only be taken up. Whereas conventional breeding takes lot of time. It's a very slow process. वो जल्दी नहीं होता है, उसको टाइम लगता है होने में. और वो इतना ज्यादा टाइम ले सकता है, लाइक इट कुड टेक इयर्स और डिकेड्स, लाइक 10 years टू स्टेबलाइज अ ट्रेट इन द जनरेशन. Whereas genetic engineering is a fast process, we can see the introduction of the trait in next generation itself. Conventional breeding is very limited. So here, whatever breeding you are doing, you can do only in the same, similar, or closely related organisms or plants. Whereas if we talk about genetic engineering, we can do it across species. Like example, I want some genes which is not there in plants, which is there in some other organism. I can try to take it. But the thing is, with that, I need to give the specific promoter, which is going to help the gene to express. That's it. But I can allow a molecule which is not supposed to be present in plant can also be delivered into a plant by the help of genetic engineering. That is feasible. So conventional breeding, what are the major examples which we see nowadays? It is hybrid maize, disease-resistant wheat, improved varieties of rice. Whereas genetic engineering, the examples are BT cotton, golden rice, drought, tolerant maize, CRISPR-edited crops. So these are the examples. Now, when we say conventional breeding, why there is a need to go for genetic engineering? Because there are lots of limitations of conventional breeding. Those limitations include time consuming, as mentioned. It takes more time to complete the process. And precision is very low. It is not specific. Precision talks about specificity. Whether I am able to generate a specific trait or not. So apart from that specific trait, you should understand that conventional breeding, I will also get the other traits. Maybe the other traits are not desirable. I want a trait from a plant that the flower should be blue in color. But apart from that, I'm also getting other traits that the trees will be short, the trees will not be able to withstand pest, withstand the. So I need to see which part is beneficial, because conventional breeding will not have specificity to choose a specific trait. It's limited in genetic scope. Genetic diversity is also very reduced because, as I said, we can only do between similar organisms or similar crops, similar plants. We cannot go beyond species. Environmental influence is there. Now, I'm trying to do a hybridization, but whether that hybrid is going to be allowed, is going to be fitting with the environment or not. Environment plays a very important note where your hybrid is going to survive or not. Unpredictable outcomes. So sometimes, as I mentioned, that you want a particular desirable trait, but with that you're also getting some undesirable traits. So that is a limitation of conventional breeding. So that brings us again to the importance of genetic engineering, because here we talk about precision, here we talk about faster crop improvement, expanded possibilities in terms of genes. So I can increase my horizon, I can do anything. It's beyond imagination that what genetic engineering can do. Enhanced resistance to pest, to herbicide, tolerance to stress like abiotic stress, which talks about climate change, drought conditions, too much of rain, everything it should be withstanding. Enhancement of the nutrition by using biofortification can also be achieved. Improved yield and productivity, which serves the uh, motto of food security, because we know to uh, food is something which we cannot compromise on. Post harvest improvements, like we can increase the shelf life of particular product, we can make sure that the process of ripening is slow so that it looks fresh for a longer time period. Sustainability and environmental benefits. So apart from making it, see, if you introduce a genetic engine GM crop, it is going to be sustainable. It is not something which is not sustainable. It can keep on generating new generations of it. And in the end, environment is going to benefit. Because we are reducing the use of how environment benefits, chemicals are less used, pesticides are less used, we are trying to get more and more crops out of it. So end of the day, environment is going to be benefited. Now, when we say genetic engineering, we have certain uh, techniques which helps us perform this process. So the first one is cloning a gene, or we also say it to be recombinant DNA technology. So what happens here? So we take a gene of interest and insert that into a vector to replicate and express it in the host cells. So basically there is a DNA segment. सो एक डीएनए की सेगमेंट है, उसको आपको एक प्लांट में डालना है. तो प्लांट में डालने के पहले हमें उसको एम्पलीफाई करना होता है. तो उसको हम ऐसे ओपनली नहीं डाल सकते हैं, उसको हमें एक वेक्टर के अंदर घुसा के डालना होगा ताकि उसकी दोनों साइड सील्ड हो जाए. Because if the ends are open, there are something called nucleases. These are basically enzymes which will chop your DNA. So there will be nothing left. So this DNA is something which is having the information that, okay, the color of the seed should be this, or the gene which is going to give me resistance to pest. This is the information which my insert is having. And I have a vector. So the insert is going to fit in the vector. It's like a jigsaw puzzle. Okay, they will fit in. Now, once they fit in, this molecule, it's a segment of DNA, it replicates. So you get an amplified DNA part of it. And that is going to be expressed in your host cells. So like this, something which is not there in a particular organism, you can make it sure that that particular gene you can transfer by the cloning mechanism. Another one was agrobacterium mediated transformation. So there is something specific soil bacteria. Huh? So the agricultural land, they have a soil, and in that soil we also see a specific bacteria which grows. That is called agrobacterium tumefaciens. So here we use this bacteria to deliver my DNA instead of plasmid. Gene cloning, we were using plasmids, okay? But here we are using a bacteria. So in that bacteria, I will put my DNA, and that bacteria will take the DNA to the nearby plants by infection. So this is by transformation through agrobacterium tumefaciens. Another one is gene gun, which is also called biolistics. So by the name itself, you can think that it's something which is like going to shoot something, right? So here we have DNA, which is present on a metal. What type of metal we are talking about? Gold or tungsten. So when these DNAs are put on these metals, they are bombarded onto plant cells with the help of air pressure. So that air is from helium. Which gas is there for the air pressure? That is helium. Okay. So that's how the gene gun is used to introduce some specific DNA into a cell. Okay. Other than that, we have CRISPR-Cas9 system. Do you know about CRISPR-Cas9? Can anyone tell me what CRISPR-Cas9 is? Previous session also you heard it, right? What is CRISPR-Cas9? आपको इम्यूनिटी पता है? हेलो
हाँ मैडम.
सर ऑडियंस आई वुड लाइक टू इंटरैक्ट.
ऑडियंस तो अनम्यूटेड है.
Okay. Khushbu Gupta is saying gene editing. Yeah, it's a technique which is used for gene editing. But do you know how it came? From where it came? See, it's a mechanism which was used by bacteria. Okay. So, like we have an immune system. हमारे पास अपनी एक इम्यूनिटी है जो हमें कोई भी इन्फेक्शन से बचाती है. उसका नॉर्मल काम ही है. ऐसे बैक्टीरिया के पास इम्यूनिटी नहीं है जैसे हमारे पास इम्यून सिस्टम. बैक्टीरिया एक सिंगल सेल है. सो उसको भी तो इन्फेक्शन होता है, लाइक वायरसेस इन्फेक्ट्स बैक्टीरिया. सो हाउ दे यूज टू get rid of it? So what they had this CRISPR-Cas9 system. Okay, so this was their immunity. So when a virus used to infect a bacteria, they use this system to chop the viral DNA. So it cannot get integrated into the bacteria and do any harm. Is it clear?
Yes,
Make
madam.
sense? Yes. So that is actually nature. Nature has given that property to bacteria. But now we, in the field of genetic engineering, we are using it. How we are using it? Same way. So some viruses are going to infect, like you talk about TMV or you talk about those viruses which infect your papaya. Now, I need to remove it. What I can use the CRISPR-Cas9 system. I can give them the code that, oh, go bind to the DNA, which is to the viral DNA. And then it will go and bind and it will cut the viral DNA. So what will happen? That virus can no longer infect my crop. That can be used in the genetic engineering technique to help you treat infections, help you get rid of diseases and other things as well. So when we say improvisations of crops in agriculture sector, we have a very important area which talks about GMOs. So GMOs has become the common term consumers and popular media use to describe foods which are created by the help, or they are bought in by the help of genetic engineering. So how we generate GMOs? There are three important steps. Basically, you identify the particular gene which you want, which is important for a particular trait. So as I mentioned, you can talk about the quality, you can talk about resistance, anything. So I want my plant to be this pest resistant. I will select a gene which will produce something which is not going to allow my pest to grow or to infect. So that is a gene. That information has to be given. So first, try to identify that particular information which is going to give that particular trait to your plant. Now, next is copy that information from the organism that has the trait. So naturally, my organism is organism B, which does not have the trait, but organism A has a trait. What will I do? I will try to take the trait from organism A and put it in B. I'm not taking the entire organism A and mixing with an organism B. No. What I'm taking the trait 1 of organism A and putting that trait 1, which is not there in my organism B. That is via the process of copying. And now the insertion of the information is happened into the DNA of another organism. So how you can do it? You can do it with uh, natural infection modes like bacteria. Why we are using natural? Because naturally they have some specific uh, property that they can infect a particular cell. So we need not put any uh, hard and fast rule. Oh, I should give something that it infects only these cells. No, bacteria itself has an ability that they will infect this cells. So they will infect, but they will not harm. What they will do, they will give you the resistant gene. Now that resistant gene can be taken up by your normal fruit, and the normal fruit now becomes a pest resistant fruit. So this slide is basically talking about the GMOs, research, review, and regulation, because when we say something is modified genetically, we need to have pass a lot of regulatory and reviews, and regulatory committees needs to verify that particular organism is for the benefit of the society and not any way going to harm the society. So on an average, GMOs take almost a huge time, like 13 years, and a huge money to be invested. Time and money is something which is very important over here, because GMOs cannot be produced within one day, and they cannot be produced with a very less amount. As I mentioned, these processes, they are very expensive and they need lot of specializations. So when you talk about modifying an organism at the gene level with specific traits, it costs you a lot. So the regulatory process alone, even if you make a product or a crop, it will still take 5 to 7 years to pass the regulations, because government needs to ensure that it is going to be good for the society, not harming anything, not harming the environment also. Okay, so it is safe to grow. Crop growth is the same as non-GMO varieties. It is not going to overuse or deteriorate the quality of soil. Crop exhibits intended characteristics like insect resistance. So what I'm trying, why a government should allow a GMO to be there when a normal crop is there? So some added advantage is there, like I'm going to reduce the use of chemicals, I'm going to reduce the use of fertilizers. If I prove that, then governments will help us in going through the regulatory aspects. Safe to the environment, like it's safe for soil, air, and water. It is nowhere going to harm the environment. Safe for beneficial insects and habitats. It is not going to harm the normal floras. Safe to eat, same nutrient as the non-GMO crops, no new dietary allergens. So it should also make sure that none of the GMOs which is going to be approved by the regulatory authorities are having any allergic side effects. Okay. So since 1992, more than 40 government agencies have been got approvals for GMO foods, feed and cultivation. In many countries, multiple agencies are involved in the regulations, and around 67 countries have now formally adopted biotech crops for food, feed, and cultivation. This report is around 2017. So, USDA gives you the approval that it is safe to grow. IPA gives you the approval that it is safe for the environment, and FDA gives you the approval that it's safe to eat. So we are not only talking about eating, we are also looking into whether it is safe to grow and safe for the environment. All the three aspects needs to be kept in check when we talk about any genetically modified organism. Coming to the timeline, we see that we begin with the crop fortifications used like breeding. Cross breedings were happening between different species. Then people started gaining interest when we talk about genetic variation in the late 1940s and 50s. Then the copying of genes happened for the desired traits, like to produce the GMOs. In somewhere 1980s, the first agricultural GMO was introduced into the marketplace in 90s. GM squash and soybeans become commercially available in the US in the year somewhere 1995, and cotton and corn become commercially available in the US markets. After that, we have papaya, canola, alfalfa and sugar beets, and then we have potatoes, and finally we are having apples by the year 2017. So coming to Indian scenario, let's see how India has come up with GMOs. So BT cotton, we all know, is now there, and it's approved, government approved, and farmers are using it. So BT cotton was engineered to produce a toxin. What is a toxin? It kills another organism. So toxin is something which is normally produced by a organism to kill another organism. Okay. So here it is by Bacillus thuringiensis, which kills bollworms. It's a major cotton pest. Now you see, as I said, genetic engineering gives you the scope to go beyond species, right? So I'm using a toxin which is produced by a bacteria to control a bollworm. How efficient it is, because normally a plant will not have it. It is only produced by a bacteria. So I'm taking the help of a bacteria which produces it. I'm taking the gene, giving it to my plant, and now my plant is producing it. Now when it is producing it, it is capable of resisting those bollworms, which is a major cotton pest, because of which, the yield and the quality of cotton was highly affected. So its introduction in India reduced pesticide use, lowered the costs, and initially increased yields, benefiting farmers economically. But with time, what happened? These got resistance. Now what happened? Another pink bollworm came by resistance. So they were now resistant to this toxin. And monoculture practices were reducing the diversity of the crop. So, see, when we say we are utilizing the modern technology, we should also know that evolution is also happening at the same pace. So if I am giving an alternative to something, I'm making it an anti-microbial, I should know that that will develop some resistance with the help of evolution within the time course. And that's where the genetic engineering needs to be cross-checked again and again. So now there is a problem that they are not able to produce good yields like before. Another one is Briti brinjal in India. So BT brinjal not happening right now. It's right now it's in a hold. It was produced by introducing a gene from Bacillus thuringiensis again, which enables the plant to produce a toxic protein, which is against the pest, but it is safer for human consumption. So it is not important. So it is very important that, when we talk about Briti brinjal and all, we should see that those toxins which they are producing are not going to harm humans, because ultimately the consumers are humans. So it should be safe for human consumption and it should not harm them. Field trials showed reduced pesticide use. Again, if we reduce the use of pesticides, we are going to reduce the cost and yields are going to be higher, offering good benefits to the farmers. However, there were a lot of concerns arose which about biodiversity loss, gene flow to wild relatives, and the long-term impact of using, modified brinjals, genetically modified brinjals. There was public opposition and there were some regulatory cautions which lead to the ban of on BT brinjal in India in the year 2010, though Bangladesh later adopted it successfully. So India is not utilizing it, but Bangladesh it is still happening. Another one is golden rice in Asia. Now, golden rice, why it is was there was a need for it. It was used to develop vitamin A deficiency was there in the normal rice varieties. So, to combat that vitamin A deficiency, a widespread health issue was seen in the Asian population, which causing blindness and immune response and immune system is also weakened. So, what they did, they inserted genes which enable the rice to produce beta carotene in the grain. So this production of beta carotene, scientists created a nutritionally enhanced staple food. So this beta carotene production was helping deal with the vitamin A deficiency. So golden rice demonstrates how the use of genetic engineering can help also address malnutrition. So not only the you know resistance to pest, we can also use genetic engineering to improvise the nutritional quality of the food or the grains which we use in our day-to-day life. But again, there are some social and regulatory hurdles which it is still facing, but in some parts of Asia it is already in use. And another one is flavor saver tomato. You can see a picture over here where you see an ordinary tomato and a gene silenced tomato. So you see ordinary tomato after like 20 days and 45 days, it is spoiled. It is full ripened and it is spoiled. So when we talk about export and import, it reduces, the quality. It does not maintain the quality to be shipped or to be exported. But when we use gene silencing mechanisms where we have used antisense RNA, so it is something which was going and blocking the ripening enzyme. So now when the ripening enzyme was blocked, what happened? It was fresh for a longer time period. So you can see a comparison of 45 days where we see that the 45 days gene silenced ones are still fresh. compared to the ordinary ones, where ordinary ones after 20 days only were looking pale and dull and they were over ripened. So flavor saver tomato was developed in the US in 1994, and it was the first genetically engineered food crop approved for human consumption. Scientists use antisense technology to suppress this enzyme. That's what I was talking about. So this antisense RNA was used to suppress the enzyme polygalacturonase, which was actually responsible for softening of the fruit. Thereby, if you decrease the production of this enzyme, what happens? The process of ripening is delayed, and it further increases the shelf life. Despite its success, the flavor saver tomato faced challenges in production cost, lower yields, and consumer skepticism about GM foods use. Another one is drought tolerant maize in the African region. So, water efficient maize from Africa, WEMA, project was developed for maize varieties with enhanced tolerance to drought conditions. This was basically done to help farmers cope up with the environmental issues. These crops provided better yields under water scarce condition, like when the now a very important problem, or a very important aspect of agriculture is water level. Groundwater level is below, very below. So water scarce condition, how to deal it? So this improved food security for smallholder farmers in places where drought conditions are much more. Despite its success, challenges remain in ensuring accessibility and affordability. See, just producing a genetic engineered crop is not enough. It should be accessible and affordable to people, because I'm trying to solve a issue which is linked to society and to farmers. If my farmers are not having accessibility to it and are not uh, you know um, having the cost to bear, then it is of no use. So something needs to be looked at that level where it becomes more affordable to people. Another one is CRISPR edited rice for blast resistance. So, rice blast fungus is one of the most destructive diseases, which is seen to affect rice varieties worldwide. Using the CRISPR-Cas9 technology, what happened? Scientists edited rice genes to enhance resistance against this pathogen with high precision. The approach reduces reliance on the use of fungicides and other chemicals and fertilizers, like although still in research and regulatory phase. So it is still not, in the market, because still research is going on, that, as I mentioned, it should be safe for consumption. Okay, it should not have any impact on other flora and fauna of the environment. So it is in the phases where these things are being checked. The regulatory authorities approve it, representing that yes, CRISPR as edited rice is a cutting edge use of genetic engineering, showing how modern tools can be used to deliver targeted, efficient, and potentially non GMO solutions for crew improvement. So, global GMO usage, we talk about so much about genetic engineering and the products which are produced from it. So, over 190 million hectares of GM crops are cultivated globally. So, you can see how many people over the world are using it and producing. Soybean, maize, cotton, canola account for the majority of the classes of the GMO crops which are being produced. USA being the largest adopter with GM maize and soybean covering most farmlands. Brazil and Argentina, major producers of GM soybean and maize. Canada being the GM canola dominates. And what about India? So in India, currently only BT cotton is officially approved for commercial cultivation. BT cotton covers around 90% of India's cotton land usage. making India one of the largest cotton producers globally. Whereas if we look into the production scale, the cotton production has declined in recent years compared to 2013-14 due to the resistance pest generation and the management issues. Both are very important. And two other genetically modified ones like BT brinjal and GM mustard have been developed but are still facing regulatory check holds and public opposition. So it is still on hold for coming into the market and being used by the farmers. So safety is very important. Whenever you talk about any genetic modification, make sure it is safe to eat. There is an organization which gives any GM foods the approval that it is safe for consumption. The WHO and FAO state that approved GM foods are as safe as conventional foods for consumption. Despite this, surveys show over 50% of consumers in Europe remain skeptical about the safety issues. So though government gives the approvals, they do validate that it is almost same like the conventional food, still people are not ready to accept it. In India, public opposition led to a suspension of BT brinjal in 2010. Already I have discussed previously about it. Despite scientific evidence of reduced pesticide use if you have BT brinjals. Environmental risk. Genetic engineering can have unintended ecological consequences. That is the major reason where regulatory offices take 5 to 7 years to approve a GM crop. A GM crop cannot be approved in 1 month or 1 year. It goes through a lot of scrutiny, lot of regulations. Then only the organizations are going to give it approval to be used. BT cotton in India initially reduced pesticide use by up to 40%, but within a decade, secondary pests like pink bollworm resurged, forcing farmers back to chemical sprays. Herbicide tolerant GM crops in the US have contributed to the rise of super weeds. Now, it's also like, AMR, antimicrobial resistance. You take a lot of antibiotics, what happens? You develop resistance. The same way, you give them one, they are getting over it and becoming more resistant. So, somewhere it is again forcing the superweed zone over 60 million acres of farmland requiring stronger usage of herbicides. The studies are showing high risk of gene flow from GM crops to wild relatives, potentially affecting biodiversity. So this is modern technology. We can use it to the best, but we should also make sure that it is not going to harm the safety and environment. Accessibility, as mentioned, since these involve lot of R&D, lot of scrutiny, lot of regulations, it is costly. No doubt it is highly costly. GM crops can increase yields by 15 to 25% and reduce pesticide costs by 20 to 30%, benefit large scale farmers. But for small scale farmers, it is difficult because they have very small lands and they cannot afford the cost of the seeds. However, the seed costs are significantly higher. In India, BT cotton seeds cost 2 to 3 times more than the conventional varieties. So, end of the day, it's all about the profits, right? Patents and corporate control mean smallholder farmers often depend on annual seed purchases, raising ethical concerns about farmers' autonomy in the process. Another important thing is public perception. 2024 global survey found that only 37% of consumers worldwide are willing to eat GM foods, even if proven safe. Though WHO, FAO are giving approval that they are safe to eat, but still people are not, convinced to eat it. In Asia, acceptance is higher when GM crops address issues like malnutrition, as I talked about golden rice. But skepticism remains strong in the areas of Europe and parts of Africa. Ethical debates are there, which talks about unnaturalness. So that's true. I already said this is something which is not naturally produced. We are making it, we are creating it, with many communities viewing GMOs as tampering with the nature. So they have their own belief systems which inhibit them for using it. To conclude the session, I would just point on the important points where genetic engineering offering precision, speed, expanded scope compared to conventional breeding. Globally, GMOs are mainstream in agriculture, especially in the Americas, covering millions of hectares with soybean, maize, and cotton. In contrast, we see that India remains highly restrictive in the use of GMOs, and the approval also being on hold by the regulatory authorities. Success of GM crops does not only depend on science itself, but also it is depending on upon the regulatory authorities, the accessibility to people, and the trust of public. Genetic engineering should be seen as a complement to traditional methods, helping address urgent global issues like food security, malnutrition, and climate change. So all these needs to be taken care when we talk about genetic engineering. So with that, I'll stop my session for today. I'll conclude and I'm open for discussion if any is there.
Thank you so much, madam.
Thank you, sir.
Anyone have any query? Anyone is there? If you have any query, you can ask to Sata, madam.